Good day, friends. Fat to Fit is here to help you lose fat, gain muscles, and stay healthier like never before. Hello, and welcome to the fifth chapter on the free course on food, nutrition, and health. Today, we're going to talk about proteins. There are few aspects of proteins that we would cover in this module: types of protein and quality of proteins, which would include important terms like biological value, PDCAAS and DIAAS, which is protein digestibility corrected amino acid scores, and digestible indispensable amino acid scores. So let us first talk about the different types of amino acids that makes up proteins. Proteins are made up of building blocks called amino acids. There are about 20 different amino acids that are linked together to form various proteins. The biochemical activity of proteins is characterized by their individual structure, size, and shape. These factors are determined by the sequence and characteristic of constituent amino acids. These amino acids are segregated into two types: essential amino acids and non-essential amino acids. Essential amino acids are those amino acids which the human body cannot produce, and hence they must be part of our diet. When you eat protein, it is broken down into constituent amino acids, which are then used to help your body with various processes such as building muscles and regulating immune system. The 9 essential amino acids are leucine, lysine, isoleucine, valine, methionine, threonine, phenylalanine, and tryptophan. Now, non-essential amino acids. Non-essential amino acids, don't let their name fool you. Non-essential amino acids fill the essential role. The reason why they call non-essential is because our body can make them, and their dietary requirement is not essential at all. There are 11 non-essential amino acids: arginine, glutamine, tyrosine, cysteine, glycine, proline, serine, ornithine, alanine, asparagine, and aspartate. In children, arginine, histidine, cysteine, glycine, tyrosine, glutamine, and proline are also considered to be essential because children are unable to make enough to meet their dietary needs. These are referred to as conditionally essential. There are also maybe certain disease conditions during adult life when a particular amino acid becomes conditionally essential. Now, protein from animal sources have complete amino acid profile. These sources includes meat, fish, eggs, milk, and cheese. These are high quality proteins as they contain all the essential amino acids necessary for building proteins and muscles. For most of us, low fat options of these foods are preferred as some can be high in saturated fat. In most diet, different proteins tend to complement each other in their amino acid pattern. So when two foods providing vegetable proteins are eaten at a given meal, such as cereal and pulses, the amino acids of one protein may compensate the limitation of the other, resulting in the combination of higher biological value. This is known as the complementary action of protein. For example, rice is low in lysine and high in methionine, while beans are low in methionine and high in lysine. Hence, when they are eaten together, they yield quality protein. Thus, if vegetarians and vegan eat a variety of vegetable proteins in combination, there is no reason why the quality of protein cannot be as good in the diet as comprising of meat, milk, fish, egg or other foods that contain the animal protein. Good sources of plant protein includes pulses, nuts, paneer, quinoa, mycoproteins, soya. There are also small amount of grains. Now we come to an interesting aspect of protein quality. There are many questions regarding the quality of plant protein. Most people assume that only animal products such as meat, fish and eggs can provide humans with high quality protein. But also numerous plants contain plenty of proteins. Each food contains a different level of protein. Besides, all amino acids are found in varying proportion in individual foods, regardless of whether they are animal or plant origin. These differences result in the food that are more convenient to supplying us with protein and some that are less suitable. There are some defined methods for determining whether a food quality is high in protein. Let us illustrate few of them like the biological value, DIAAS and PDCAAS. The biological value. The biological value indicates how much of the protein eaten can theoretically be converted into body's own protein. Biological value is a measure of the proportion of absorbed protein from a food which becomes incorporated into the proteins of the organism. It captures how readily the digested protein can be used in protein synthesis in the cells of the organisms. Proteins are the major source of nitrogen in food. Biological value assumes the protein is the only source of nitrogen and measures the proportion of this nitrogen absorbed by the body, which is then excreted. The remainder must have been incorporated into the protein of the organism's body. A ratio of nitrogen incorporated into the body over the nitrogen absorbed give a measure of the protein usability or the biological value. The higher the content of the essential amino acids, the more efficient the use of corresponding food proteins for the synthesis of the body's own protein, and the higher is the biological value. The reference value for the biological value is egg, which has a biological value of 100. However, this does not imply that egg is the best possible source of protein. By combining different foods, you can even reach a biological value of over 100. Here, it is only important that the combined foods complement each other in the amino acids profile, the essential amino acids that we are talking about. The limitation of biological values that looks at the protein efficiency from what is available in the body. In order to overcome this, we have PDCAAS and DIAAS. Now we know what biological value is. However, it is not enough to only know the value of the theoretical intake of food protein. Even more important is how much of the protein in the diet the body can actually absorb during the digestion and further use it on. To measure this, we have two methods, PDCAAS and DIAAS, or the protein digestibility corrected amino acid scores and the digestible indispensable amino acid scores. Both scores describe the quality of proteins based on the human protein requirement and the ability to digest them. For a long time, the PDCAAS was considered a method of choice to measure a protein quality. In the meantime, however, DIAAS is considered more accurate measure of the actual uptake of the individual amino acids. The DIAAS assess protein quality based on the true ileal digestibility of each essential amino acids. Now, ileum is a part of the large intestine for those who are not aware. It gives a value for each of the 9 essential amino acids. On the other hand, the PDCAAs reflect the fecal digestibility of the protein and only indicates the value of the limiting amino acids. In the next chapter, we would cover protein digestion, how much protein is adequate, and the role of important amino acids. If you have any queries or concerns, please feel free to post your queries in the comment and I would get back to you as soon as I can. I hope you found the information valuable and I would like to thank you for watching it. Take care of yourself and your family and I'll see you in my next video.